Today we're going to interview Christina Towers. She's an assistant professor and researcher at Salk Institute. And Christina works with cancer and is currently studying the recycling mechanisms of cancer cells. Welcome, Christy.
Hello.
First of all, we would like to thank you for sharing your time and experience with us in this interview. For sure, it's going to be very inspiring, and we have a lot of questions to you.
So, we know that your research line is related with cancer cells and their recycling pathways, autophagy. But from this study, you find out that cancer cells can adapt to autophagy inhibition. Do you already know some pathway that explains this cancer cell mechanism when the autophagy inhibition happens?
Yeah, fantastic question. So the research that I did has been looking at autophagy, which is a cellular recycling process. We think cancer cells are really addicted to these pathways because these recycling processes allow cancer cells to generate their own nutrients. And tumors are really harsh environments, so there's not a lot of oxygen or blood supply. So you can imagine that a cell that can just recycle its own nutrients has an advantage and can continue to grow, right? But what we've shown is that if you block these pathways, cells are so dependent on these autophagy pathways that they just find another mechanism, another way to get the same thing done. So what my lab is focused on now is trying to understand exactly the question you asked, which is what are these mechanisms that cells can use to get around such an important process? So we do have some ideas of what those could be, and we've discovered that by using cancer cell lines that we grow in what we call 2D culture, so growing them in plastic dishes, and we've discovered that, for example, the paper that I think you're referencing, we asked, well, how can these cells help maintain their protein homeostasis, right? So if autophagy is important for breaking down proteins, if they can't do autophagy, well, how else can they break down their proteins? And so we showed that the cells can instead use, they can upregulate a master transcriptional regulator called NRF2, which directly regulates all of these proteasomes, which is another way to break down proteins, right? So we're sort of finding these workaround mechanisms that the cells can use. We've also shown that cells can do alternative pathways to maintain mitochondrial homeostasis. So mitochondria are these large organelles in the cell. They're important for energy, for the creation of ATP, and they're important for a lot of other things as well, but they're really these large organelles, and the only way that a cell can get rid of a damaged mitochondria is through this autophagy pathway. So we asked the question, well, if our cells can't do autophagy, the cells, that we've blocked autophagy in, well, how can they be maintaining mitochondrial homeostasis? And so we've discovered that they're using, again, this sort of alternative pathway where they're taking bits and pieces of mitochondria. Instead of taking a whole mitochondria and putting it into an autophagosome to be degraded, instead they're taking bits and pieces of mitochondria called mitochondrial-derived vesicles, and they're getting those to be degraded, sort of circumventing, again, the sort of traditional pathway. So those are some of the mechanisms that we already know, but I think for sure there are going to be a lot of other mechanisms. But I also think one of the downsides to the work that we've done so far has really been exclusively in cancer cell lines that are growing in two-dimensional cultures on plastic with all the nutrients they could ever want in the world. And that's just not really a physiologically relevant context. It's a great context to study molecular biology and to study signaling, but it's not anywhere near the conditions that would be seen in an animal or in a person. So now we're trying to build systems where we can study these same questions, but in more physiologically relevant systems. So we're using mouse models now, but we're also using 3D tissue culture models where we have sort of organoids that are growing in culture, and they have multiple cell types as part of them, and they're grown in conditions that are much more relevant to what you might see in an animal. So we're still pursuing those questions and trying to identify new mechanisms, but also the mechanisms that we have already identified, ask how relevant are those in real conditions?
Well, in your last published papers, you investigate the relation between these recycling mechanisms and cancer, right? So how can these findings help in the cancer therapy? In cancer, actually.
Yeah, that's really, again, good question. So as I said, autophagy is a recycling pathway, and cancer cells can become really addicted or dependent on these recycling pathways. And so right now, across the world, there are lots of clinical trials that are being done to try to target this pathway. And some of those trials are going well, but some are not, and that's because patients are responding, which means their tumors are shrinking when we block autophagy, which is great, but then their tumors start to grow again, even though we're blocking the same pathways. So that says that the tumors are becoming resistant to these therapies, and the patients are relapsing, which means that even though their tumors went away, now their tumors are coming back. And they're no longer responding to therapy, so their tumors are just still growing. But what we're working on here is sort of leveraging our knowledge that we can get from understanding the basic science, right? Can we understand these alternative pathways that cells can use to, say, degrade mitochondria, right? If we know what those mechanisms are, can we identify drug therapies to target those mechanisms? And then we could combine that therapy with an autophagy inhibitor, which would prevent the patients from becoming resistant. Another thing that our work does is, instead of trying to find therapies to block the tumors, is can we actually predict which patients will respond to the therapies we already have, right? So if we know that we have these autophagy inhibitors, they block the pathway, but we know there are these alternative pathways the cells can use, well, maybe we could screen patients ahead of time and say, oh, patient 1, you don't have any of those alternative pathways, so you should get the autophagy inhibitor. Oh, but patient 2, you have all of these other pathways in your tumor cells, so why don't we try putting you on a completely different therapy? The autophagy inhibitor is not going to work for you. And so now we're not wasting precious months giving people therapies that are never going to work, and we can get them on the therapies that will cure their cancer before it gets too bad.
It's very interesting. It's very interesting, the mechanisms.
Yeah.
What are the biggest difficulties you deal with in cancer research?
The biggest difficulties that we deal with in cancer research, broadly speaking, I would say the biggest issue in cancer is that every single person's cancer is different. So it's very different than, say, like a coronavirus, right? Where the virus is identical in every single person because the virus is what's causing the disease. But with cancer, because cancer comes from your own cells and every single person is different, that means that every single person's tumor is different. So instead of having to find treatments for one disease, like we're doing with COVID-19, we now have to essentially find a treatment for every single person because they're all different, right? So we try to group them, right? We try to say, okay, all of these patients have the same breast cancer, so we'll treat them all the same. All these patients have the same pancreatic cancer, so we'll treat them the same. But I think what the last 10 years has shown us is that just doesn't work. That's just not true. You can take 50 women that all have breast cancer, but every single one of them, their breast cancer is slightly different, right? And they all respond to different treatments. They all have different mutations. They have different pathways that are activated, and they need to be treated as like 50 different diseases instead of all the same disease. So it makes the problem exponentially bigger than trying to, say, treat a virus or a bacterial infection, right? So I think, the age of, since we've been able to sequence the human genome, now we can sequence all of these tumors and we can really understand all of these different mutations. And that helps us group tumors accordingly, right? To say, okay, this group of patients, you all have a mutation in the BRAF gene, you have a mutation in P53. You know, you can group them based off of what their genomes look like. And that helps us. But I think even now, even with all that sequencing data, we still have patients, you know, that all have the same mutations even, but we give them the same drugs and they don't respond the same. One patient's completely cured and the next patient is, you know, dies from their disease 3 weeks later, right? So what's different between those two patients? We still really don't know. So I think that's probably hands down one of the biggest issues that we have in cancer biology right now. And I would say the other one is exactly what we're working on, which is resistance to therapies, right? It's that the tumors are changing every time we treat them. The tumors change and they evolve. And you kill most of the tumor, but then there are a few cells that are resistant to that drug. And so now those few cells survive. And even though you shrunk the tumor, a year later, now those 10 cells that survived, now they've grown out to become the whole tumor, right? And now you have this super aggressive tumor that's resistant to every drug that we throw at it. And so, yeah, I think, like we were saying earlier, cancer cells are just really smart, you know, and they are under immense pressure to survive, which means that they will find ways to do that. They will activate pathways that they weren't using before. They will turn on genes that they weren't using before, and they will find a way to survive. And so it just becomes really, really hard.
Yeah.
Thank you so much, Christy. I really appreciate it.
Thank you.
It was my pleasure.
That's so great.
We love the talk.
Yeah. All right, just email me if you have any other questions that come up.
Okay.
Okay.
All right.
Thank you so much.
Bye.
Bye, Christy.
Bye.